Okay. Hello, everyone. My name is Brianne Palmer, and I'm a fifth-year PhD student at San Diego State and UC Davis. And today I'm going to be talking to you about why you as botanists should care about biocrusts. And in short, you should care about them because they're very cute. They're also like mini forests. Plants love or hate them, and I'll explain more about this later. And most importantly, I think understanding biocrusts will change the way that you look at the world. So in this talk, first, I will attempt to define what a biocrust is and give you some background on the key biocrust organisms. I will also explain what they do and how this impacts the plants that we know and love. And finally, I will explain what I've been working on during my PhD. So in general, most of the biocrust research in the United States occurs throughout the Western deserts. So I put this map up just to familiarize you with the research I'm going to share today. There are four main deserts: the Great Basin, the Sonoran, the Mojave, and the Chihuahuan. And then my research is not in the desert. It's in the California grasslands in coastal Southern California. So now jumping into what exactly are biocrusts and why are they so interesting? And to understand this, we have to go all the way back to the formation of life on Earth. So single-celled cyanobacteria were some of the first life forms in the ocean, and they were responsible for oxygenating Earth's atmosphere via photosynthesis. But it's also hypothesized that cyanobacteria and algae were the first organisms to colonize the land. They were able to move from the water to the soil, where there are a few nutrients to survive, by photosynthesizing. After colonizing the soil, the cyanos improved soil stability and added nutrients to the soil, which allowed land plants to eventually grow. We can still see cyanobacteria colonization today, and this is considered the first step in biocrust development or succession. So for example, this is a photo I took of a cyanobacteria in the Mojave Desert. Some microbial ecologists might call this a biofilm, but it is aggregating the soil particles together, so we consider it biocrust. And I got lucky and snagged this picture right after a rainstorm. So we can see all these little green squiggles coming up to the soil surface. Those are cyanobacteria, and they move to the surface to take advantage of the moisture and to photosynthesize. And if we look closer, we might see a consortium of cyanobacteria, but common in these early successional crusts is the genus Microcoleus. And as you can see in this video, Microcoleus moves around in its own blanket of slime called EPS, or the exopolysaccharide matrix. And I like to think of EPS as cyanobacteria glue. It binds the soil particles together, and it also provides a safe place for other microbes to live in. But Microcoleus isn't the only important biocrust cyanobacteria. Another genera common in these crusts is nostoc, or the dark overlord. Nostoc generally colonizes after the filamentous and cyanoglue producing bacteria and provides a whole new set of ecosystem functions. Nostoc can be associated with lichens, but can also form crusts without fungal associates. And it's one of the organisms responsible for forming the dark pigmented soil crusts that are fairly typical on the Colorado Plateau, like in this figure here. So I really like this photo because it shows just how large of an area biocrust can cover if just given the right space and the right climate. And it also provides like this nice little home for these bunch grasses. Nostoc is a heterocystis bacteria, meaning it creates these special cells here that are deprived of oxygen. And this allows Nostoc and other heterocystis bacteria to fix atmospheric nitrogen, much like rhizobia on legumes. Nostoc also provides additional stability, desiccation tolerance, and adds photosynthetic capacity to the biocrust. So this figure from Young et al. 2018 describes the differences in photosynthesis activity in cyanobacterial-dominated biocrusts, and I think it's really informative about the role that cyanobacteria play in biocrust development and function. So this crust is about 4 centimeters deep. In the top layer, this is this gelatinous nostoc layer, and right below that is the filamentous cyanobacteria and green algae. So nostoc and the filamentous cyanobacteria both provide nitrogen fixation and stability. But as we move deeper into the soil, there's more filamentous cyanobacteria that aren't close enough to the surface to photosynthesize, but they're still producing that EPS and binding the soil aggregates together. So all of the soil binding and making of soil nutrients paves the way for other organisms to colonize the biocrust, like lichens. So lichens are a symbiosis between cyanobacteria, or an algae species, with a fungus. And in biocrust, it seems that lichens are a little bit pickier about where and when they choose to colonize. If we think of biocrust like a forest, the cyanobacteria would be that early successional Doug fir coming in fast after a disturbance, where the lichen is more like a Western hemlock. It's very picky about where it grows, and it's often a late successional species. And you can see in these photos that lichens on biocrust come in all different shapes, sizes, and colors, but they all perform fairly similar ecosystem functions. And also within the biocrust, we can see that lichens are incredibly diverse and have different structures. Some are flush with the soil, some are more bulging, and they also all grow together with multiple lichen species growing right next to each other. So if you want to impress your friends on your next hike, here are some of the common lichen genera in California. I'm not a lichenologist, and I'm still trying to learn lichen ID, but there is a free field guide published online called A Field Guide to Biological Soil Crusts of Western US Drylands. And it provides a key and a lot of information about how to identify lichens and mosses. So in San Diego, in Southern California, this is the most common lichen genera that I see while hiking around in the coastal sage scrub. Cladonia has these really tall fruiting bodies, and they're little cups. Calema is common in biocrusts around the world, and it's another one of those organisms that give desert biocrusts that really dark black color, and it's a nitrogen fixer as well. Peltula is common in the Mojave Desert, and again, it's a nitrogen fixer, and it's often confused with Placidium. They look very similar, they have similar fruiting bodies, but Placidium is a slightly different color. They're often grow right next to each other, and they're both nitrogen fixers. And then my favorite lichen genera is Sora. It has this really beautiful rose color that makes it easy to identify, especially after a rain, and it's also very common in the Mojave Desert. And there are so many more lichen species in biocrust, hundreds if not thousands, and I identified these lichens just based on my own photos and basic ID skills, but I highly recommend you check out this book available online. And it's also important to understand that all of these lichen species grow with each other, and they also grow with other biocrust species like mosses and cyanobacteria. So as I mentioned, lichens provide many different ecosystem functions. They fix nitrogen with the help of cyanobacteria. They increase soil moisture, but importantly, they also change surface roughness. So surface roughness is really important in drylands because it reduces the amount of water leaving a system. Sometimes biocrusts are classified by the surface roughness. For example, we would call this a smooth crust, where the crust itself is only 0 to 1 centimeters tall. And this is a valuable measurement to take in the field to understand the role of biocrusts in the ecosystem. So biocrusts that are nearly flush with the soil like this provide other benefits like biogeochemical cycling, but they don't have the best geometry for capturing water. And these crusts are really common in hyper-arid deserts or disturbed areas, like where I took this photo on San Clemente Island. So just to show this graphically, when the rain hits the crust on the soil, there's just not a lot of structure to contain it, and it runs off. These crusts are typically made up of cyanobacteria and algae because lichens and mosses provide a little bit more structure for the water. So the second type of crust structure is a Rugos crust, and these are 1 to 3 centimeters tall, so slightly taller than the smooth crust. And just this little bit of variation in the soil surface helps it capture more water. And these crusts are more common in less arid areas like the Mojave Desert. So when it rains, water hits the surface, and some of this water is trapped in the gaps created by the biocrust, and the tiny ravines in the crust force the water to meander across the soil surface and allows for more water infiltration. These are also dominated by cyanobacteria, but the clumps of lichens and mosses are what add to that rugosity and really help capture the water. And the final crust structure I want to bring to your attention is the pinnacled crusts, and these can be 5 to 15 centimeters tall. Some crusts in this area were almost up to my kneecap, so they can get very tall if left undisturbed. And these are really common in the Colorado Plateau region. Cyanobacteria are key to structuring these crusts, but they also have up to 40% cover of lichens and mosses. So when water moves through these tiny desert skyscrapers, it provides more opportunities for the soil to drink up that water as it moves across the landscape. It can get trapped here, feed some of the plants and the other organisms that rely on water in the desert. And the final component of biocrust I want to introduce you to are bryophytes. These include liverworts and mosses. And if you saw Jenna's talk in this series a few months ago, she explained how and why some desert mosses, which are common in biocrust, are so drought resistant. And there are actually over 300 described bryophyte species in biocrust, and they're usually the last organism to colonize. So this first photo here is a liverwort. It took me a long time to figure out what this was in the field, but what we're seeing with these black lines is actually the sheaths, the underside of the leaves. So the leaves are closed, and once we get this wet, they open and they have these brilliant green leaves. The second photo is also a dry moss, and it's hosting some really pretty plants. And the dry moss can be really difficult to identify in the field because of the dark color. It can look like a cyanobacteria or even a lichen. But if you look at it closer and even touch it, it has a nice soft texture like a moss. And then this third photo is cyanobacteria, algae, and mosses all growing together, forming this really nice green biofilm. And it's found growing on a clay soil in between two bunch grasses. And this final photo is just an example of a plant seedling, and there's another little seed here germinating through a moss biocrust. So just like the lichens, I just want to give you a little bit of background of some common bryophytes in North America. And again, I'm leaning heavily on this field guide to North American biocrusts. And like I said before, mosses can be difficult to identify when they're dry, so it's best to identify them after a rain, or if you bring a spray bottle to the field and just spritz it and let them brighten up a little bit. So Bryum argentum is a ubiquitous moss found on soil and other substrates. And this is probably the most common moss that I've seen in Southern California. Centrichia ruralis is also common in the deserts of North America, but it's less common where I live in the coastal environments. But it has these distinctive awns and this star-like structure. So when it rehydrates, it like twists up, and it's known for its fast response to rewetting. And then finally, this is Athamelina hyalina, which is a liverwort that ranges across California and into parts of the Great Basin. And I've seen this one growing near vernal pools and really moist shaded habitats. So just to summarize all the different crust types and their functions, I want to highlight this paper by Nicole Petrusiak from 2013. And she measured the different functions of each biocrust type, particularly nitrogen fixation, carbon fixation, and soil stability. So on the x-axis here, we have the three different categories I talked about: the algae, cyanobacterial crust, lichens, and mosses. And she broke it down even further into more specific crust types. But generally, this moves from the least developed crust to the most developed crust. And on the y-axis here, this is nitrogen fixation. So as we move from the least developed to the most developed, nitrogen fixation increases. And the same story holds true for carbon fixation. Again, moving from these really light algal crusts to the more moss-dominated crusts, there's an increase in carbon fixation. There is some variability in the amount of carbon and nitrogen fixed by each crust type, simply due to the types of organisms within the crust. Some lichens and mosses fix more nutrients than others, so it's important to understand each component of the crust community. And interestingly, biocrust type had a really strong effect on soil aggregate stability. So this is how stable are the soils. It can also be measured by using that cyanoglue, the EPS component. And what Nicole found was that the early crust, this incipital algae crust, which only has a little bit of cyanobacteria, was not very good at aggregating the soil. But as we move up to all the other different soil types or soil crust types, you can see that they all have a high ability to bind those soil aggregates together. So in total, it's estimated that biocrusts sequester more carbon than they emit. It's estimated that they fix about 4.9 picograms of carbon annually. They also fix more nitrogen than they emit, releasing some of that fixed nitrogen into the soil, which may find its way into plant roots and other microbes. So how do all of these organisms and their functions impact plant communities, particularly biocrust structure and biogeochemical cycling? So there are a couple ways biocrusts can interact directly with plants. The first is by inhibiting their growth. So it may not be the best of news if you are a botanist who wants plants to grow, but some of these biocrusts form this thick layer and can make it difficult for seeds to germinate. However, when seeds do germinate, they're in this nice, safe place, flush with nutrients, higher soil moisture, and they have a safe place to grow. So the facilitation and inhibition of plants was covered in a meta-analysis by Carrie Havrilla, and I just want to briefly go through her results now because I think they're really interesting, especially as you consider the role of biocrusts in plant monitoring and management. So just to orient you to this, the plots, the biocrust type is on the x-axis, and the y-axis is the effect size. So a positive effect size means that there is an overall positive influence of biocrust on the measured variable compared to the same experiment done in bare soil. So overall, plants performed worse in lichen-dominated biocrust compared to the bare soil, and there wasn't a huge effect by other crust types, despite mosses and lichens playing similar roles in biogeochemical cycling and soil stability. Generally, lichen biocrusts are more inhibitive to seed germination compared to bare soils, and this can be a blessing if you're using biocrusts to inhibit non-native germination like cheatgrass in the Great Basin, but it can be a hindrance in establishing other plants. However, when seeds are able to germinate through a lichen-dominated or mixed biocrust, there is significantly more growth compared to bare soil. All of the biocrust types had a similar influence on plant survival, so it likely depends on the plant and the environment more so than the crust type when it comes to survival. And lastly, moss and mixed biocrust did promote more plant cover than plants grown in bare soil. Biocrusts and plants can also interact indirectly through nutrient cycling. As I mentioned earlier, biocrusts are key nitrogen fixers in dry lands, but now researchers are trying to understand if plants can actually get the nitrogen that is fixed by biocrust. So is this a sharing relationship or a competitive one? One of the hypotheses is the fungal loop hypothesis, which is the idea that there are these fungal networks connecting biocrusts to the plants which allow them to share nutrients, similar to mycorrhizae in plant roots helping plants share nutrients. There are currently multiple groups studying this using stable isotope tracing, but the existence and effectiveness of the fungal loop depends largely on the plant species, the biocrust species, and the climate. So in dry grasslands, for example, the fungal loop was present, but it only benefited the plant growth and seemed to have a negligible effect on biocrust growth. And because of their roles in nutrient cycling and their potential benefits to vegetation, biocrusts have lately been the focus of restoration. So biocrusts can be restored a couple of ways. First, they can be salvaged from sites slated for disturbance, like construction sites. And the salvaged biocrust can be transplanted directly into the field and eventually reestablished. So in this photo here, all of this dark soil in the background is covered with biocrust. And you can simply remove parts of this biocrust into this disturbed area and it will eventually reestablish. Researchers are also looking into ways to grow biocrusts in the greenhouse, just like we do for plants. But this has presented some challenges. When the greenhouses, I'm sorry, my headphones just went off. Can you guys still hear me?
You blipped for a second, but I can hear you again now.
Okay, cool. My headphones just died. Okay, so researchers are also looking at ways to grow biocrusts in the greenhouse, but this has presented some challenges. Mainly when greenhouse biocrusts are transplanted into the field, they often fail to establish. These are similar problems that we face with plants when doing restoration. You have to sort of ameliorate the conditions. The greenhouse conditions are very nice and they don't like being moved to the harsh desert. So in this photo here, this is just a normal greenhouse tray filled with soil. And you can see that it's really easy to grow biocrusts in the field or in the greenhouse, but again, it's difficult to transplant them from this stage into the field. So in terms of biocrusts, botany, and the broader ecosystem, there's still a lot we don't know about how biocrusts interact with plants, and it may be different in every ecosystem. We do know that biocrusts and plants co-evolved, and this is likely influencing modern ecosystem dynamics. And I also think that conservation and restoration efforts should consider both biocrusts and plants, since these communities are clearly interacting, they're living in the same environment, and saving one may have drastic effects on all trophic levels in the ecosystem. But in order to properly conserve biocrusts, we must first understand the threats and how they recover from disturbance. So this is where my PhD research comes in. There are a variety of threats to biocrust biodiversity and function, including tramplings by humans and livestock. We can limit this by avoiding hiking on top of biocrusts and rethinking how we graze cattle in highly crusted areas. Recreation and off-road vehicle use are also threats in the desert, where ATVs and even tanks have driven off the road, and it can take decades or longer for the biocrust to recover. So in this photo here, there's biocrusts all across the desert. Someone drove their truck, and it's going to take decades to centuries for biocrusts to fill in these gaps. And like all ecosystems, climate change is a big threat to biocrusts due to changing precipitation patterns and persistent droughts. And that's connected to the disturbance that I have spent my PhD studying, and that's biocrust response to fire. This is particularly important as fires are increasing in severity and frequency. Everywhere around the West right now is on fire, and it's important to know how all these communities may respond. So in order to understand how biocrusts respond to fire, I performed a meta-analysis with all of the studies about biocrusts and fire from 1975 to 2019. And I found 237 unique experiments from 21 papers. And the meta-analysis revealed that overall fire reduced the cover of biocrusts, especially cyanobacteria and algae, which is shown in this figure here. So all of my data has a negative response, but cyanobacteria and algae have a much more negative response, especially compared to the other biocrust types. However, the strength of this response was modulated by time since fire. So generally, as the time since fire increased, the biocrust cover also increased. So here on the x-axis, we have time since fire in months, ranging from 0 months to 400 months. And for all of the biocrust types, except for cyanobacteria and algae, we actually see an increase in biocrust cover. So fires initially are bad, but over time, we are gradually seeing biocrust cover return to like pre-fire levels. So next, I wanted to understand how biocrusts in California recover after fire, especially California grasslands, which are understudied in the biocrust literature, as most studies tend to focus on deserts and shrublands. So I did most of this work on San Clemente Island, which is a naval base off the coast of San Diego. It's the southernmost Channel Island. And the plateaus on the island have a very diverse grassland, where the majority of the moisture is coastal fog. And these grasslands harbor a diverse biocrust community. Since it's a drier grassland, there are these vast patches of soil between the grasses, and these interspaces have a lot of biocrusts. The biocrusts can be found growing directly adjacent to the plants or even underneath them. San Clemente also has a recent and well-documented history of fires and established plots, which allowed a good setting to understand biocrust response to fire. So on San Clemente, I have 3 sites, and just to orient you to this figure, the open squares are plots where there is prescribed burn, and the darker squares are areas where there was no burn. And sites 1 and 2 were burned in a prescribed fire in 2012, and the same plots were burned again in 2017. Site 3A was burned in a wildfire in 2012, and then again in a prescribed burn in 2017. And then I added new plots here in site 3, and these were only burned in a wildfire in 2012. And then each of the prescribed burns only occurred within a 10-meter square area, so we're not burning the whole landscape here, just small plots. This is an example of what one of the plots look like. This is at site 1. So each plot is 10 meters squared, and I believe this picture was taken a year after the 2017 fire. So each plot is 10 meters squared, divided into 4 quadrats. Within each quadrat, we placed Whittemire frames and measured percent cover of biocrust and plants within the frame. I then collected biocrust from outside of the frame for lab analyses and DNA. So in the lab, we measured chlorophyll, which is indicative of photosynthetic biomass. We also measured nitrogen fixation. So inside this desiccator, there are tiny biocrust samples, which are then exposed to an atmosphere with a nitrogen isotope. We could then calculate how much of the labeled atmospheric nitrogen was fixed by the biocrust using a mass spectrometer. And finally, we extracted EPS, or that cyano glue that can bind the soil aggregates together using a weak acid extraction. And from each site in treatment, I also collected biocrusts for microbial sampling. I extracted DNA and sequenced 4 samples from each site treatment combination for a total of 24 samples. These were sequenced using shotgun metagenomic sequencing, which is a way of sequencing microbial communities to not only see what microbes are present, but also the potential functions they might be performing. So to answer my first question, does fire change biocrust cover? The answer is yes. Biocrust cover was completely different in the burn plots compared to the controls, just not in the way that we expected. So for my meta-analysis, I expected fire to reduce biocrust cover, especially cyanobacteria and algae. But here you can see site on the x-axis and total cover on the y. Red is the burn plots and blue is the control plots. So across all sites in all years, there was more biocrust cover in the burn plots compared to the controls. Additionally, most of the biocrust we saw on the island was dominated by cyanobacteria and algae, with only a few patches of lichens and mosses. So this may be different in the meta-analysis results because many of the studies in the meta-analysis looked at fire effects 5 to 10 years after the fire, which may have been enough time to see that decline in cyanobacteria as lichens and mosses gradually increase. And this study occurred 1 to 3 years after the 2017 fire, so we aren't seeing the immediate effects of the fire, nor are we seeing the long-term effects. But this provides valuable insight into that in-between phase. And we do have some data supporting the idea that eventually biocrust cover will decline when we look at the correlation between average plant cover and average biocrust cover. So there's a negative correlation between biocrust cover and plant cover only within the burned plots, indicating that the plants and biocrust may be competing for space or light or other resources, and the plants generally win. Therefore, after a fire, there are less plants and more space available for the biocrust to quickly colonize. So this is an example of what the quadrats looked like in the burn to control plots. So this one was burned, and you can see there's a lot more empty space between the plants, and that's where the biocrust is growing. And this grass here is actually Cyphopulchra, and the fires were intended to increase Cyphopulchra abundance. This also led to the creation of more biocrust habitat. In the control plots, the plants are much more dense. There's a thick thatch layer. These are mostly invasive annuals like bromus and avena, and they create little space for the biocrust to grow and little light for them to photosynthesize. Another possible reason we saw greater biocrust cover is because the biocrusts in this ecosystem are either highly resistant or highly resilient to fire. So many organisms within the biocrust are resistant to heat stress in the form of drought tolerance. And it's possible that this heat avoidance strategy helps them survive the fire. Additionally, some microbes are motile and may live deeper in the soil to avoid the destructive heat of the fire. This is the same video of the Cyanobacteria microcoleus I showed at the beginning of the talk. And in the context of fire, this ability to move might allow it to escape the heat. And another possible mechanism is simply fast recolonization. There are millions of microbes floating in the air, and since these plots were located near undisturbed areas, these areas may act as a source to quickly recolonize the bare space after a fire before the plants were able to regrow. So we didn't see what we expected with biocrust cover, but maybe the functions were also undeterred by the fire. And we did find that fire didn't change the amount of chlorophyll or photosynthetic biomass in either the sites or either year. So not only is there greater biocrust cover after a fire, the biocrusts that are there are just as photosynthetically active as their unburned counterparts. Additionally, fire didn't change the overall concentration of EPS, or that cyano glue, in sites 2 and 3. There was, however, more EPS in the burned plots in site 1 in both 2018 and 2019. And this is really surprising because we expect EPS to increase with successional age, but we're assuming that these burn plots are in a lower successional state than the control plots. And we also didn't see this effect in site 2, which has a similar soil type. It's only 1 kilometer away and experienced the same burns. So this is a really strange and interesting result. And finally, the amount of nitrogen fixed was also unaffected by fire. We saw a lot of variability in nitrogen fixation between sites and years, likely due to changes in the microbial metabolism during incubation. However, the samples collected from the burn plots in 2019 had greater nitrogen fixation than the controls. So similar to what we saw with EPS. And again, this is just a very strange result. It could be because 2019 was a wet year compared to 2018, which could have influenced the microbial community. But why we're only seeing it in 1 site out of 3 is something to ponder.
That doesn't make sense. Oh. Huh. That is weird.
All right, I guess, yeah, let's wait a minute and see if she comes back on again.
Maybe you can text her or call her, Amy, if you have her number to let her know.
Unfortunately, I don't. I can only email her.
No, it's just some bird.
Let's just be patient for a second. I would think she would know, but.
Yeah.
She texted and said the storm knocked out her internet.
Oh dear.
Oh boy. Storm. Oh. That's too bad.
Well, if it's knocked out completely, then I don't know who that was who just spoke. Can you text her back and see if there's any way she can join, or is it?
Here she is.
Yeah,
Hi.
there you are. Okay,
All
great.
right, there's like a freak lightning storm and it knocked out our power. It's back down.
Cool, you're back. That's all we care about. Are you ready to pick up?
Yeah, just let me share my screen again.
Great.
I need permission to share my screen.
So where are you that you're getting a storm?
San Diego. It was so
Really?
unexpected. It came out
Wow.
of nowhere.
Huh.
Congratulations on having a storm.
That storm, yeah, a storm that was coming up on the west side of Mexico. Maybe.
There we go.
Okay.
Okay, it's all good. Hopefully the power stays on.
All right. Microvirus. No. Huh. So what? Methylbacterium. Okay, that's fungus.
Okay, can you all see my screen?
Yes.
Okay.
Can everybody just mute yourselves again, please?
Okay, so I left off talking about what microbes we saw in the burn sequences. And we saw similar microbes in the burn plots as we did in the controls. Streptomyces is still really common. but Geodermatophilus is more common, and nostoc actually decrease in relative abundance. And this is important because Geodermatophilus is not a cyanobacteria, but it is a microbe known to survive really harsh environments like exposed rocks, and it's pretty common in biocrusts from hot and arid environments. But the differences in the most common genera don't provide the whole picture regarding the microbial community. So we looked at the overall community composition with the permanova shown here on an MDS plot. So the closer together the points are, the more similar the communities are. And from this, we can see, based on the overlapping of the ovals, that there's no difference between the communities for each fire type. So essentially, there are similar microbes across both fire types and the control. The control is in blue, the prescribed fire is in red, and the wildfire is in yellow. So the wildfire had a larger variation in the community composition, but this was taken into account with the permanova, and there was still no significant difference in the community composition. However, when we perform the same analysis for the functional genes, we see that there is a slight difference in the functional gene composition between the control in blue and the wildfire in yellow. So these sites may not differ taxonomically, but there could be slight changes in function. This suggests there may be unaccounted diversity below the taxonomic levels we included in this analysis, such as at the species or the strain level. And additionally, terrestrial cyanobacteria and other soil microbes are very underrepresented in microbiome databases, so it's difficult to fully understand the scope of the diversity. So to understand exactly how biocrusts are different functionally, I looked into the gene categories in a database that basically uses an algorithm to match DNA sequences to known sequences of microbial functions. And I specifically looked at chlorophyll, EPS, and nitrogen fixation, since those are the genes I measured in the lab as well. And I didn't see any difference in the relative abundance of chlorophyll genes, which matched our lab measurements. Additionally, with EPS genes, there is no difference between any of the treatments, which matched our lab results. And there is no difference in the abundance of nitrogen fixation genes either, which again matches what we measured in the lab. Some gene groups, however, were more abundant in the wildfire plots. For example, there are more genes for phospholipids, which are important for membrane formation and cell communication. And there were more genes related to cell division. So maybe the microbes in the wildfire plots are doing a few things differently than the control and prescribed fires. Other gene groups were more abundant in the controls, such as chemotaxis genes, which are related to cell mobility. Invasion and resistance genes, perhaps because they're older and they have more microbes around trying to harm them, and so they need more genes to combat that. And there are more genes relating to viruses because just like us, biocrusts get viruses too. So now I'm interested in exploring new questions for my final chapter of my PhD. I'm interested if biocrust microbes can survive fire by escaping the heat. Does fire change which microbes and their functions are active? And how does this affect the broader ecosystem, especially if they're more severe and more frequent? So to answer the first question, I collected biocrusts in the field and I burned them in the lab, shown in this video here. So in this video, this is the biocrust on top and it's sitting on top of this coffee mug full of soil. And it's either burned at 450 degrees Celsius or 600 degrees to simulate heat from a low intensity or a high intensity fire. And we monitored soil temperature at different depths using thermocouples. We also had a model of what we expected soil temperature to be at different depths shown here. So as you can see in the red and the orange and the yellow, the first few centimeters of the fire is very hot. But if we just go a few centimeters below the soil surface, it drops to a level that could be survivable by many microbes. I also included the thermocouple data here. And the thermocouple data actually matched our model fairly accurately. It was about a centimeter off. So either we need to tweak the model or the biocrust is actually like adding an extra layer of protection to those deeper soil layers. And then after I burned, I took a little soil core from the top all the way to the bottom, and using that soil core, I cultured cyanobacteria and algae that could survive at each depth of the soil profile. So this is an example of 10 of the hundreds of plates that I have in the growth chamber. This particular group was burned at 600 degrees, And you can see the top centimeter that was exposed directly to the fire had no growth. But even as shallow as 2 centimeters, we'd had photosynthetically active growth all the way down to the bottom of the mug. So this is a heat map of all the samples. The blue rectangles show where there was growth and the red rectangles show where there wasn't. And across all of the samples and all the treatments, there's actually more growth at the 2 and 3 centimeters than I expected. And theoretically, these microbes living at that shallow of a depth could survive the fire and potentially form a new biocrust. So right now I'm currently sequencing the DNA of these cultures to determine what microbes are able to grow. And I will also be sequencing the RNA to understand exactly what microbes survive the fire and sort of what they're doing, which ones are active. And this will allow me to better understand the functions they're performing. And then from here, researchers need to understand what's happening under field conditions. This is a very small scale experiment. It works in the lab, but how it actually applies to real world fire is something that we still need to look at. So to conclude, I have a few take-home messages about what's been a whirlwind talk through biocrust ecology. First, I hope it's clear that biocrusts are important for the biodiversity and function. As one of the first life forms on land, they're instrumental in our understanding of terrestrial ecology. Second, the interactions with plants are complex, and like many things in ecology, we need to have collaborations between plant people, soil people, and micro people to really understand how these spheres interact. And third, understanding just how biocrusts respond to disturbance is necessary to conserve them. So climate change and fire are real threats to these communities along with countless others. And since we're just scraping the surface about what biocrusts do for ecosystems, we also need to keep working on making sure these communities stay intact. So I hope I convinced you that biocrusts are cute and charismatic communities that need to be included in management and biological monitoring due to their importance to the ecosystem. So if this picture doesn't convince you that they're cute, I don't really know what does. I also want you to realize that they're like little ecosystems within themselves, and they have a large effect on other organisms around them, particularly plants, which have a complicated relationship with biocrusts, whether they're trying to germinate through that thick lichen layer, or they're benefiting from the nutrients and soil stability. And finally, I do hope that understanding how biocrusts work and what they're doing changes the way that you look at the world. My first experience with biocrusts was camping with my parents in Arches National Park in the early 2000s. And it was the first time someone told me that soil was alive. And 15 years later, I'm getting a PhD. So studying biocrusts really is life-changing. With that, I'd like to thank my advisor, David at SDSU, and Valerie, who hosted me in her lab at UC Davis, as well as Rebecca, who co-authored the meta-analysis with me, Nicole, who's been helping me understand cyanobacteria and algae, as well as Don Lawson and John Keeley, who orchestrated the research on San Clemente, my field assistants and lab mates through the years who went out and collected samples with me, as well as all of my funding sources who covered sequencing and travel costs. This is a brief list of references, which is by no means extensive. If you want more materials, feel free to reach out to me. And with that, I'll leave you with this image of what it's like to hike with me. I'm sure many botanists can relate, and I will take any questions.